the histone acetylation in peripheral white blood cells and in the brain of mice. It also restored normal mitochondrial function in cortical neurons and rescued the sociability defect in the KAT6B heterozygous mice. And then ALCA treatment by itself, so not VPA, rescued the spatial recognition memory defect and strategy learning defect. And this is quite remarkable. And the part that I haven't told you about is our assessment of some KAT6B mutations. So you are all familiar with the fact that a large number of KAT6B mutations have been reported in the KAT6B gene, and they cause two different disorders, chiefly Say-Barber-Biesager-Young-Simpson syndrome here in blue, and the genitopatellar syndrome mutations in red, and some intermediate in green. So what we know there is how the gene is mutated and that this causes the disorders, but we don't know what the functional consequences of each of these mutations is. And here it's relevant that the gene is transcribed into mRNA, and mRNA is translated into protein, and then proteins show a range of functions. And I just want to stress to this audience that every single one of these mutations need to be assessed for their functional consequences. And we don't have the capacity to assess them all, but we've assessed 8 of them, and 8 distributed along the whole length of the KAT6B gene and protein. And Hannah Wanyai generated these specific Say-Barber-Biesager-Young-Simpson syndrome mutation in a human cell line, the HEC293 T cells. And then Maria Bergamasco examined them. And here I'm showing you the mRNA levels. So these are KAT6B mRNA levels in the cell lines that have either no mutations, so the control here in white, or these specific mutations inserted into their genome. And you can see that we have 6 cell lines with 6 mutations that have reduced levels of KAT6B mRNA, and 2 that have normal or even slightly higher than normal levels. The 2 that have normal levels are in this last coding exon here, this last big exon. The mutations in the more proximal exons all caused some form of reduction in KAT6B mRNA. And this is a phenomenon that is called nonsense-mediated RNA decay. So the machinery notices that there is a mutation in the RNA, and then it gets decayed or degraded. So that's how we arrive at these lower levels. But it's important to note that these lower levels of mRNA are not 0, which means that theoretically truncated proteins or proteins with amino acid exchanges could be generated. And therefore, it's also important to look at protein function. And we look at 2 protein functions here. One is the histone acetyltransferase function that KAT6B and also KAT6A has. And we know from other work that the main target for KAT6B is histone 3 lysine 9 acetylation. And then there's a secondary target, histone 3 lysine 23 acetylation. And we see that histone 3 lysine 9 acetylation is just slightly but significantly reduced in 6 of these mutations, namely the 6 that also have mRNA level reductions, and not reduced in these 2 in the last coding exon. And histone 3 lysine 23 acetylation is reduced in the 2 first ones. So then Maria also looked at mitochondrial function, or the effect of mutations on mitochondrial function, rather. The mitochondria are the energy plants of the cell. Their output is ATP. And you can see here that surprisingly, not the 6 that were affected by histone acetylation, but these 3 central mutations only. So that's this one, and this one, caused a reduction in ATP production in the mitochondria. So I just wanted to show you this because this needs to be done for all patient mutations, we can't assume that we know how any of them works. And overall, our KAT6B work has been published, so you can read this. And I know many of you have already in this paper in the Journal of Clinical Investigation, where Maria Bergamasco is the first author. And now I want to come to KAT6A for the rest of the presentation. So KAT6A was previously known as monocytic leukemia zinc finger protein, and we've worked on this for 25 years now. And we initially studied its normal function in mice where it's required for hematopoietic stem cell development and maintenance, POX gene expression and body segment identity, TBX1 gene expression and cardiovascular development, and DLX gene expression and craniofacial development. And Perez Campo and colleagues reported on the brain development effects. And then, as you all know, Arboleda et al. And Thumb et al. Reported that heterozygous mutations in the human KAT6A gene caused intellectual disability and facial and skeletal dysmorphogenesis. So the Arboleda-Thumb syndrome. And our more recent work is, which we now are preparing for publication, is that KAT6A is modeling KAT6A disorders in KAT6A heterozygous mice, where we observed, examined the basic effects on mouse behavior and cognition, and also tested potential treatments. And so the progress was slower on the KAT6A project and then on the KAT6B project due to two reasons which are important for understanding the results. So first of all, the KAT6A heterozygous mice are small. They show a failure to thrive between birth and weaning. So weaning is when they no longer require the mother, which is at 3 weeks of age, and not all survive. In fact, 30% die on an inbred genetic background. Therefore, it was slow to build cohorts of mice for behavioral tests. And then the second and more important problem is that the behavioral phenotype of the KAT6A heterozygous mice is milder than that of the KAT6B heterozygous mice. So we have a smaller difference between the wild-type controls and the KAT6A heterozygous mice, which make all results less clear. It's more difficult to establish whether there is a treatment benefit. But we have now completed this study. And so I'll start first with our work on assessing the functional effect of some of the mutations. So you know that there are very many, and I can just stress again, the functional effect of these in terms of the protein function need to be established. And we've done this for 6 mutations. So Hannah again made these mutation in the human HEC293T cells, and then Chesley and Samantha analyzed them. And again, we tried to spread the mutations that we analyzed across the gene. And you can see here at the mRNA levels that the mutations here, these 5 mutations, so these here resulted, or these 4 mutations resulted in a significant reduction, 5 mutations in a significant reduction of mRNA, and this last one, the one that is in the last coding exon, had normal mRNA levels of KAT6A. So that's again indicative of nonsense-mediated RNA decay in these transcripts that have mutations in these proximal exons, but not in the final exon. A expected mechanism, but again, we reduce not to 0, but to about 50% of wild type here in white. And that means theoretically these mutant proteins can be produced at some level. Then we looked at protein function, namely histone acetylation. And here the main target is histone 3 lysine 23 acetylation. And that was reduced in these cell lines with these 4 mutations, these 4 more proximal mutations, but not the last 2. So even though we have a reduction in mRNA levels here, this one doesn't seem to have a reduction in histone acetylation, which already by itself is interesting. So, and that sort of result is important if you're thinking of using treatments that might increase histone acetylation, which would not be appropriate for individuals who have this mutation, because histone acetylation doesn't seem to be reduced. Okay, so just briefly regarding our KAT6A mutant mice, how do they relate to the mutations that are found in the Abuelidas thumb syndrome? So here I grayed out the mutations that we looked at, and I'm showing now the exons that we have deleted to produce the KAT6A mutant mice. So these are exons 4 to 8. And you can see here, these are RNA sequencing reads over individual exons in the KAT6A gene. And you can see here that the deleted exons are gone in the homozygous mutant cells, and they are reduced, their messenger RNA here is reduced in the heterozygous. And then Hava examined the effect on histone acetylation in mouse brain. And you can see that the KAT6A heterozygous mice, which I will show now always in red, compared to the wild type, which I will show in blue, have a reduction in histone 3 lysine 23 acetylation. And Maria examined the peripheral white blood cells, and here again you can see a reduction in B cells and these two populations of T cells. So the peripheral white blood cells could be used to assess this in individuals with KAT6A mutations as well. So then Maria further isolated neural stem cells from embryonic day 12 embryos from the dorsal telencephalon, which is the precursor of the cerebral cortex, and grew them in culture as these floating colonies. They're called neurospheres, and examined histone acetylation again. And these are now, we are here now comparing homozygous mutants versus wild type, and can see that histone 3 lysine 3 acetylation is reduced, and histone 3 lysine 9 acetylation is unchanged, and lysine 14 acetylation is also unchanged. Samantha then also isolated neural stem cells and examined their proliferation and differentiation in vitro. So here we have all three genotypes, the wild types in blue, the homozygous mutants in green. You can see they proliferate substantially less over, this is over 5 weeks in culture, each week has a passage. And the heterozygous cells don't grow slower than the wild types. And indeed, if we look just at passage 5 here, we can see that they may even grow a little bit faster than the wild types, but the homozygous certainly grow slower. All three genotypes have a similar percentage of actual stem cells. They are SOX2 positive cells. And then when Samantha differentiated the cells in vitro and looked at the content of neurons, astrocytes, and oligodendrocytes, we can see that the KAT6A heterozygous here in red and the homozygous in green produce a lower percentage of neurons compared to the wild type cells. All genotypes produce similar percentages of astrocytes and similar percentages of oligodendrocytes. And then the last in vitro system that Sam looked at were cortical neurons from 4-week-old KAT6A heterozygous mice versus wild type. And here I'm showing pictures, and they look similar. The proportion of neurons in these cultures is similar. The number of neurites per cell, so these are these projections here, are similar. We distinguish the primary neurites that emanate directly from the cell body, and the secondary, which emanate from the primary, and then the tertiary, which are branch off from the secondary neurites. And the number is all the same, and the length is also the same. So with that, I'd like to come to our behavioral assessment of the mice. So this is the battery of mouse behavioral tests that we established, and I will take you through these here highlighted in red, and they will provide an assessment of motor coordination and sensory perception, physical parameters, vocalization, visual acuity, motor function and strength with several tests, somatosensation, so the ability to feel things, general activity levels and locomotor activity, anxiety-related behavior, and then several cognitive tests for spatial working memory and recognition memory, declarative memory, recognition memory, spatial strategy learning and memory, and then social interaction. And hopefully, and you will see that it's not, our findings are not clear-cut in all cases. So we first looked at a developmental series. This is from birth to day 21 of age, and you can see this is the body weight development. So the wild type, the KAT6A heterozygous mice have in red develop much more slowly and remain small throughout their life, despite the fact that they are born with a normal weight at birth. And here you can see a KAT6A heterozygous mouse and its litter mate control. So this is a sibling. Look how much bigger it is. And the developmental milestones were largely normal, but I'm showing you two here which were different. So mice are born with their eyelids closed and they open their eyelids after birth, so about 2 weeks after birth. And the KAT6A heterozygous mice opened their eyes 3 days later than the controls. But they showed an earlier development for negative geotaxis. This is where the mouse is placed with the head down on an incline, and eventually they develop this ability to turn around and face upwards with the head, and the KAT6A mice do that much earlier. Other developmental milestones were largely normal, such as pivoting, surface writing, cliff avoidance, auditory startle, tactile startle, and bar holding was a little bit later in the KAT6A heterozygous mice. The grip strength relative to body weight is the same. Both genotypes avoid a visual cliff, so they stay on the solid ground, which means that they can see. And here in this adhesive removal test, we place a sticky tape on the paws of the mouse, and this test assesses whether A, the mouse can feel the sticky tape, and B, whether it can remove it, whether it has the fine motor to remove it. And that also appeared normal for the KAT6A heterozygous mice. We tested their motor function and strength on the rotorod. So here the rod turns and the mouse has to walk with it. If it cartwheels like that, there will be a penalty for it. So you can see that the KAT6A heterozygous mice perform less well on this test than the KAT6A wild-type mice, which may relate also to the fact that the bar is rather thick and the KAT6A heterozygous mice are quite a bit smaller. We then assessed vocalization in the mice. Mice vocalize, emit ultrasonic vocalization, so we can't hear them, but they can. And we tested them in 2 different situations. One is the postnatal maternal separation-induced vocalization. So the pup is separated from mom and then it calls to mom, and we can measure the calls. And the other one is courtship-induced male vocalization. So the male mice sing to the female mice. And what you can see here is that the KAT6A heterozygous pups at postnatal day 4 and 12 emit fewer calls. And the male KAT6A heterozygous spend less time singing to the females and also emit fewer calls during that time. Here I have 3 tests that assess the level of anxiety, and also these 2 tests also assess the level of activity, so hyperactivity or lower than normal activity. So in the large open field, the distance traveled is larger in the KAT6A heterozygous mice than control, and they also travel at a higher speed. So these 2 parameters combined are interpreted as hyperactivity. The mice also traveled a substantially longer distance in the elevated zero maze, again suggesting hyperactivity. Now, in terms of anxiety in the large open field, mice prefer to stay around the periphery, but they venture out into the center, but less so, because that exposes them, and they might feel that that exposes them to aerial predators. So the KAT6A heterozygous and wild-type mice spend a similar proportion of time in the center. So this did not suggest any changes, any abnormalities with the anxiety. However, in the elevated zero maze, where the mice choose to be in this open area or in this area here enclosed by the walls, we saw that the KAT6A heterozygous mice spent significantly more time in the open compared to the wild types, and also entered the open areas more commonly. So these two tests didn't agree with each other. So we engaged a third test, the light dart box, where the mouse can choose to spend time in this clear area here, which is made of perspex, or go into this dark part of the box. And also here, the KAT6A heterozygous mice spent more time in the light area. So now I come to the cognitive tests. And this is where it becomes really quite subtle in terms of the differences, and as you will see. So in the Y maze, the mouse is placed here into the home arm, and it is allowed to examine this arm, and this other arm here is cut off. In a second session, that cut off arm is now open. This becomes the novel arm, and this the familiar arm. And wild type mice show a preference for the novel arm over the familiar arm, as measured by percentage of time spent in the two arms. The KAT6A heterozygous mice do not show this preference. And at this point, I need to introduce to you the discrimination index. For the discrimination index, we take the time exploring the novel arm and subtract the time exploring the familiar arm, and then divide the number by the total time of exploration of the two arms. And you will see that this preference of the wild type mice here shows up as a positive number in the discrimination index, so near to 0.2. And the failure to preference any of the arms shows up as a number near 0. And if there was a preference for the familiar arm, it would go further down into the negative numbers. And then we can compare these two preferences, and we see that there's a significant difference between the two genotypes. So that was the percentage of time spent in the novel arm. But we can also look at the percentage of entries. So this is a mouse that has gone into one of the arms with all 4 paws. And you can see that the wild type show with this parameter a strong preference for the novel arm. And the KAT6A heterozygous mice now also show a preference. And with the discrimination index, we see that perhaps the preference is not quite as strong, but it's sort of, the difference is borderline significant. So for us, any number here below 0.05 would be significant. And the third parameter that we can look at is the distance traveled in the novel versus the familiar arm. And you can see again, the wild type mice travel a longer distance in the novel arm than the familiar arm. And so do the KAT6A heterozygous mice, travel a longer distance. And if we look at these two preferences, we can say that the preference in the KAT6A heterozygous mice is significantly weaker than in the wild type mice. But it's not an all or nothing. This would be an all or nothing result where we have a preference in the wild type and no preference in the heterozygous mice. Here we now only have a quantitative difference, which will cause trouble when we start the treatments. In the object recognition test, the mice, as you know, as you may have heard me speak about before, are first faced with 2 familiar objects, and then in a second session are given a novel object, and the wild type mice strongly prefer the novel object over the familiar object, so they spend more time with the novel object. And the KAT6A heterozygous mice also prefer the novel object, but show a weaker preference compared to the wild type mice. Okay, so this is the last test and our most elaborate test of learning and memory. So the mice are placed on a large platform in the center, and there's bright light and noise there. And these are the aversive stimuli that the mice might want to avoid. On the periphery of the platform are 20 holes, but only one of them, the target hole, leads into a nice dark tunnel. and a nice dark chamber. And over 4 days, the mice learn how to find the target hole. And then we look at several parameters. So we have the number of holes visited. And you can see that both genotypes learn over time to visit fewer holes before they find the target hole. And the results are identical between days 1, 2, and 3. But then suddenly on day 4, the KAT6A mice spend more time visiting the target hole, visit more holes before they arrive at the target hole. Similarly, the time needed to find the target hole shows that both genotypes learn over time. They, over these days, they use less time to find the target holes on day 3 than day 1. But then on day 4, KAT6A mice use a bit more time again. So this was by itself would be difficult to interpret, but we have another clue, which is the latency to enter the target hole. So stepping down into the target hole is not quite as easy as one might think. And so because the KAT6A mice are smaller, we had to adapt the step down. We had to put in more steps, so it was easier for the smaller KAT6A mice to enter the target hole. So this is now the latency to enter the target hole, which is a combination of the time until we find the target hole plus the time until we finally disappear in the target hole. And you can see that on each of these days, the KAT6A heterozygous mice take longer to actually enter the target hole. Now that may be because they're smaller and find it difficult to enter the target hole, or because they're less bothered by the loud noise and the bright light that they experience on top of the maze. So the final parameter is the most interesting one, and this is about search strategy learning. So we distinguish here a random search in gray, a serial search in the lighter gray, and a spatial search in the black. And the random search is the mouse visits several holes before it finally, almost by chance, arrives at the target hole. The serial search is the mouse goes out to the periphery and runs along the periphery until it finds the target hole. And then this so-called spatial search is the direct approach to the target hole, either directly or we allow a mistake of plus or minus 1 hole. And what we can see here in the wild-type block over the 4 days is that the wild-type mice reduce the random search here in the darker gray and increase the spatial search, so the direct access to the hole in the black. In contrast, the KAT6A heterozygous mice do not show any changes in the strategy used. So that's quite interesting. So they do arrive at the target hole and they enter, but they don't learn the spatial strategy. They also don't seem to rely on a random strategy because they use the serial strategy quite effectively. So with this, I come to the sociability test. So here we have 3 chambers and the mice can choose. There's an aversive floor here in the center. The mice then can choose to go into a chamber with an empty cage or a chamber with a cage that has a mouse in it. And under these circumstances, the wild-type mice strongly prefer the mouse, spending time with the mouse over spending time with the empty cage. And the KAT6A heterozygous mice also prefer spending time with the mouse, but again, their preference is weaker than the wild-type preference. But you could easily see that, so now I need to prepare you for the problems that we encountered when we treated. You can easily see that if the wild-type mice performed a little bit less and the KAT6A mice performed a little bit better, there could be a random reason why, we are just missing 2 lowly performing mice, they would be similar. And that's the problem that plagued us. Okay, so Samantha then tried a large number of different treatments. And here I'm showing you the weight of just the heterozygous mice at 21 days of age. So this is our baseline here that I've talked to you about. And this is the developmental milestone cohort, which you've also seen the results of. And then we have VPA treatment and control, ALCA treatment and control, high fat versus standard pellet, Devetelact, which is a veterinary product that is very similar to Sustagen in composition. versus standard pellet and citrate versus the vehicle for citrate. And what I wanted you to take away from this graph is that the weight of the mice doesn't change. So it's not about making them grow better, making the heterozygous mice grow better. So I've shown you results for these two cohorts. I will now just talk about ALCA and its vehicle and citrate and its vehicle. And this is because the results of the other cohorts were inconclusive. So we can't really, and in some cases, uninterpretable. So for the ALCA treatment, we treated from 2 weeks of age to 4 weeks of age, once daily, with acetylcarnitine subcutaneously. And the control was a saline vehicle control also subcutaneously. And then from 4 weeks of age, we made a mesh for the mice. So this is taking their normal pellets and grinding them up to a powder and then adding water, to which we then either added acetylcarnitine, or we gave them the controls were a mesh without the acetylcarnitine. And in these following slides, I will always show the vehicle control in the solid bars and we had the expectation that the vehicle control would be like our baseline assessment. But that's also not always the case, as you will see. And then the treatment group is in the open bars. So here I'm showing histone 3, lysine 23 acetylation in the brain. And as we had in the basic assessment, KAT6A heterozygous mice have a reduction in histone 3, lysine 23 acetylation in the brain. And this is slightly increased in the ALCA-treated group, so that the ALCA-treated heterozygous are now no longer significantly different from the control mice. And we see something similar in the spleen of these mice as well. The rotarod, so what I will show you for the behavioral studies is the baseline cohort results here with a slightly gray background. So just to remind you, you saw that the KAT6A heterozygous mice were performing less well in the rotarod. So the time until they fell off the rotarod was shorter than the controls. We also see this in the ALCA-treated mice. So ALCA did not improve motor coordination or strength in these mice. The vehicle-treated mice were curiously more similar to the wild type. We have no explanation for that. So in the sociability test, we also did not see an effect of ALCA. Let me explain this. So you remember that there was that the KAT6A heterozygous mice did show a preference for the mouse over the empty cage, but their preference was weaker than that of the control. And here in our ALCA treatment group, this preference of the heterozygous mice, vehicle-treated heterozygous mice, for the mouse over the empty cage was not statistically significant, but looked as if it was there. And ALCA, the ALCA-treated heterozygous mice did not show a preference. And you can see this here in the discrimination index. However, we had such a spread that the vehicle-treated mice were not significantly different from the control mice. So in the Y maze, we encountered similar problems. So you remember in the baseline cohort, KAT6A heterozygous mice showed a preference for the novel arm compared to the familiar arm, and this preference was weaker than the preference of the wild-type mice. In our ALCA and vehicle cohort, the KAT6A heterozygous vehicle-treated mice showed a preference for the novel arm, and the ALCA-treated vehicle mice also showed a preference for the novel arm. And on the surface, all preferences in all treatment groups and genotypes look very similar. So so this poses a problem. So ALCA could improve spatial recognition memory to the point where the ALCA-treated heterozygous are no longer different from the ALCA-treated wild type or the vehicle-treated wild type, and that's the case. But the vehicle-treated heterozygous also did not show the phenotype. And in fact, if we look at the level of preference, so now this number here, we can see that the wild type, vehicle treated or ALKA treated, show a slightly reduced preference compared to our wild type in the baseline assessment. And the KAT6A heterozygous mice actually don't show much of a difference in level of preference. So that's why we concluded there's no improvement, even though these look all the same. In the object recognition memory test, we now have a clearer effect, whereby, as you remember, the heterozygous mice in the baseline cohort preferred the novel object over the familiar object, but the preference was lower than in the wild-type mice. And we see that the vehicle-treated heterozygous mice show a strong preference for the novel object over the familiar object, but the ALKA-treated heterozygous mice do not, and that can be seen here as well. Now, whether or not there is an improvement with the vehicle is something that would be very difficult to conclude from this data set. So where we did see an improvement is with hyperactivity. So here are our vehicle-treated mice in the open field where the KAT6A heterozygous spent a significantly greater distance, or traveled significantly greater distance than the KAT6A wild-type mice. And then after ALKA treatment, the KAT6A heterozygous mice travel a similar distance. We also see this improvement, a level of improvement in the novel object recognition test, where we also looked at the distance. Again, in the vehicle-treated mice, the heterozygous travel a longer distance than the controls. And in the ALKA-treated mice, they now travel a smaller distance, which is no longer different from, significantly different from the controls. So this looks more like a partial rescue, and this more like a more complete rescue of the activity levels. And then the final data that I will show you refer to the Barnes maze, which you remember we have these three strategies. The two blocks here are wild-type mice, either vehicle-treated or ALKA-treated, and they show, as we saw before, a reduction over the 4 days of the random search in the darker gray and an increase of the direct access, the spatial search to the target hole, both in the vehicle-treated group and the ALKA-treated group. Our KAT6A heterozygous vehicle mice did something similar as in the baseline. They did not increase the spatial search, but they did reduce the random search in favor of the serial search. So they are pretty good at the serial search. After ALKA treatment, however, they did increase their spatial search significantly and did reduce their random search even more significantly than in the vehicle. So this was a remarkable finding. The ALKA-treated heterozygous mice might not use the spatial search quite as much as the controls, but clearly more than if they were not treated with ALKA. All right, so with this I'd come to citrate. So here we injected citrate from 2 weeks of age to 4 weeks of age intraperitoneally, or we injected saline as a vehicle control. And then from 4 weeks of age, the mice received citrate in the drinking water or plain drinking water. And I should say that this has no effect on histone acetylation levels. I'm looking here at the activity levels, and you can see again that the saline group of heterozygous mice traveled a much greater distance in the novel object recognition test than the controls. And the citrate-treated mice were still significantly different from the controls, although a larger cohort may have shown a difference, a slight reduction. We didn't get that here with these numbers that we have. In the novel object recognition test, you recall that the novel that the KAT6A heterozygous mice preferred the novel object, but that their level of preference was weaker than in the wild-type mice. Now, in both the saline-treated group and the citrate-treated group, the heterozygous mice showed a strong preference for the novel object, similar to the preference that the wild-type mice showed. And in the discrimination index, we see now that all genotypes seem to prefer the novel object at a similar level. So this is difficult to interpret for the following reason. The wild-type mice did not reach the same level of preference as in the baseline study. So it's almost as if the wild-type mice preferred a little bit less, and the heterozygous mice preferred the novel object a little bit more. So that's why I said it may improve object recognition memory. It's a little clearer in the sociability test, where you recall in the baseline data set, the heterozygous mice preferred the mouse over the empty cage, but their level of preference was weaker than the wild-type mice. Here we clearly see that the level of preference of the heterozygous mice, both in the saline-treated group and the citrate-treated group, is similar to the wild-type, and similar to also to the preference that the wild-type mice showed in the baseline assessment. So we can say that citrate, but also its vehicle, saline IP, seem to improve sociability in the KAT6A heterozygous mice. So in summary, I want to say that KAT6A heterozygous mice display a reduction in vocalization events. They display hyperactivity, lower than normal levels of anxiety, reduced recognition memory, both spatial and contextual, a strategy, learning and memory defect, and reduced sociability. I've shown you that KAT6A heterozygous neural stem cells give rise to fewer neurons, that KAT6A heterozygous neurons cultured in vitro have a normal morphology, and that the KAT6A mice display a failure to thrive postnatally. And I haven't shown you, we also observe relatively mild anomalies in the gastrointestinal system. Treatment with ALCA amazingly improved hyperactivity and spatial strategy learning. Treatment with citrate, but weirdly also with its vehicle, the saline, improved sociability and possibly somewhat recognition memory. And with this, I'd like to acknowledge the researchers involved, in particular my long-term collaborator, Tim Thomas. Tim and I co-supervise all of our projects. Most of the data I've shown were generated by Samantha Eccles. Hannah made all the human mutations in the human cell lines, and Hava, Maria, and Chesley assessed histone acetylation levels and mRNA levels, and Charlotte genotyped the mice. We had a fruitful collaboration with Anthony Hannon, who advised us on mouse behavioral tests, and Adam Vogel, who is an expert in ultrasonic vocalization in mice, and we are very grateful for these funding bodies. Thank you very much for your attention.
Thank you very much, Dr. Voss. A quick question about vocalization. Did you see any change with ALCAR, any improvement or no change?
So we didn't test the male. We could have, you're right, we could have tested the males after ALCAR treatment, but we didn't do that. So we needed, for the ultrasonic vocalization test, Adam Vogel lent us a particular apparatus where you place the mice into and record the vocalizations. And we had that for this developmental study at the beginning of our study. And then later, when we got into the treatment phase, we would have had to get him to lend it to us again, which disrupts his own work.
Thank you.
Hi, I wanted to see if I could ask a question.